Hello, everyone. Today, I would like to show you some of the work that our group at Sunnybrook Research Institute in Toronto, Canada, have been working on, focusing on quantitative image analysis and single-cell phenotyping using imaging protein multiplexing. I will give you some examples of how we use this technology to study the characterization of breast cancer and also on work that we did in the study of immuno-oncology. Before I go into my talk, I just want to highlight why we wanted to study single cells. Long time ago, when I first studied cancer in university, cancer is pretty much a linear problem. We know that some normal cells in a tissue probably acquired a mutation, either on a tumor suppressor gene or an activation of an oncogene. These cells were then able to proliferate uncontrollably and eventually able to metastasize to nearby organs. We now understand that cancer progression is much more complicated than a linear problem. Each cell will be able to acquire and accumulate mutations during their cell divisions, and the cell that acquired a proliferative advantage will become the dominant clone, which will then be able to proceed to metastasize. Therefore, the progression is a branch process during which different cancer clones will be generated and contributing to the resulting heterogeneity. Heterogeneity not only exists in the cancer, but also in the tumor microenvironment. We know that the TME is made up of different functional cell types, immune cells, fibroblasts, and vascular cells. We also now know that these cells are not all the same. Some could be activated while others are suppressed. In addition, the spatial distribution patterns could be very different. Therefore, the TME could also be heterogeneous and highlights the importance of single-cell analysis to comprehensively identify and quantify them. In this talk, I will show some of the work that we did looking at heterogeneity in breast cancer and in the cancer immune microenvironment, particularly on the tools that we are developing for spatial analysis. The protein multiplexer that we have in the lab is called MXIF, or multiplex immunofluorescence, which is now commercially available as the cell dive imaging system. It is based on a sequential stain bleach method, whereby each round of staining will consist of a pair of Cy3 or Cy5 conjugated antibodies. In terms of the image analysis workflow, it's actually quite similar to other protein multiplexer. DAPI is imaged in every round so that we can register all the images at the end. And also there is a workflow for automated cell segmentation. So after all the analysis, we will get an output of these features of each cell together with the expression of each protein on single cells. One of the unique features of the multiplexing or the cell dive system is the preservation of the tissue section after staining, which allows us to conduct routine H&E staining of the same slide. This way, we will have both the clinical pathological tissue features and the molecular multiplex marker signatures of the same cells, which has been very valuable for our digital pathology developments. So for the first application, I will talk about our work studying biomarker expression heterogeneity in breast cancer. In the clinical pathology lab, breast cancer are classified into different grades and groups based on the morphology and the immunohistochemical staining of biomarkers of estrogen receptor, progesterone receptor, HER2/neu, which is also often confirmed with fluorescent in situ hybridization staining. Around 20 years ago, with the advances in molecular profiling technologies, breast cancers are categorized into different subtypes based on the expression profiles of certain genes. And these are luminal A, luminal B, HER2-enriched, and basal-like subtypes. There is also a stratification called the IHC surrogate subtypes, which uses IHC staining of biomarkers as a surrogate to predict the molecular subtype of the cancer. Even though it looks like the classification system are equivalent at some levels, this is actually not the case. In fact, there are a lot of evidence suggesting that discordance between the clinically defined classes and molecular subtypes. For example, up to 10% of hormonal receptor positive clinically defined breast cancers do not belong to the luminal molecular subtype. Heterogeneity in the cancer can probably contribute to some of these discordance. Before we use protein multiplexing to study heterogeneity, we wanted to know if multiplexing measurements of these breast cancer biomarkers correlated well with IHC measurements in the clinic. We did multiplexing and IHC of the same biomarkers on serial tissue sections and measured the histoscores scores as determined with multiplexing or with IHC for ER, PR, and HER2, and the percentage of positive cells of Ki67. As shown here, we found good correlations between measurements from multiplexing compared to IHC, suggesting that multiplexing measurements for these markers are good indicators of what are clinically measured. We then studied the quantification of biomarker staining with multiplexing. In this case, HER2 is shown in the upper panel. Spot 041 here is a very strongly stained HER2 positive breast cancer. But when we looked at the single cell expression level of HER2, we actually found quite a range of different levels that we detected with multiplexing, showing again the variation in biomarker expression levels. We also looked at PR. Spot 11 here is a PR positive breast cancer. However, when we look at the single cell expression levels, again, we see very large variation in the spread of signal intensities of PR as measured using multiplexing. One of the approaches that we use to classify different single cells is to use a binary cell count system, where cut point levels for each single marker was determined by comparing the positive and negative tissue controls, and applying these cut points to classify cells into positive or negative. By doing this, we get 8 groups of ER, PR, and HER2 co-expression signatures, which we called EPH. Since the MXIF or the multiplexing measured expression levels of ER is quite weak or low, we could only differentiate between the ER negative weak from ER moderately and strongly stained cells. When we use this classification system and studied the composition and distribution of single cells across different subtypes of breast cancer, we found that the triple negative subtype as shown here at the top, as expected, only expressed group 1 cells consisting of ER low, PR negative, and HER2 negative cells. The HER2 positive subtype is also made up of cells mostly with HER2 positive. For the luminal subtypes, though, we see quite some variation in terms of the type of cells with ER and PR positivity, as we can see from the different colors and the composition of EPH cells as shown here. For each TMA core that we analyzed, we also quantified the single cell expression levels of P53, P21, and P16, and their positive fractions in each core. Even though triple-negative breast cancers and HER2-positive cancers appear to be more homogeneous in terms of ER, PR, and HER2 expression, we see quite varying levels of P53 proteins and P16 proteins in these 2 subtypes. Also, we found that there are a few cases that carry P53 overexpression mutations as detected by IHC actually showed a variation or very high levels of proteins when measured using multiplexing, suggesting that when we look at single cell expression of these proteins, we can see the heterogeneous patterns, and they are not very uniform, and they are not uniform among different cancer subtypes. We studied the spatial arrangement of these cells, and this is done by quantifying co-occurrence matrices. The number of times that a certain cell type, which we call the neighboring cell types, are in the proximal neighborhood of the central cell type, in this case, cell type number 1. The size of the neighborhood can be defined by the user, and in this case, as indicated by R as radius. In most of our studies, we used a distance of 100 pixels, which correspond to 30 microns. We studied the neighborhood composition of 3 different breast cancers, 2 of which are luminal B and 1 luminal B HER2 positive cancer. From the presence of the color-coded cells, we can see that spot 135 on this upper left panel is very homogeneous, whereas spot 117 is the most heterogeneous. This is also reflected from the proportion of other EPH cell types in the neighborhood, as shown here by the bar graph. We also use Shannon equitability index to assess the diversity of the composition of the neighborhoods for these cells, and it showed that some cell types tend to be in more heterogeneous neighborhoods than others in the individual cores. Another method that we use to visualize this data is to use unsupervised clustering. When the same tissue microarray dataset was clustered with Phenograph, which is a high-dimensional clustering approach to identify clusters of single cells that are phenotypically similar in terms of their marker co-expression patterns, we found a total of 25 of these clusters, each with a very different pattern of protein marker co-expression signatures. Shown here is an example from cluster 11 on the right-hand side. Again, this demonstrates the high level of heterogeneity when we study expression signatures of single cells within each IHC-defined subtype. We have not only looked at inter-cancer heterogeneity, but within a patient, we also found high levels of variation between multiple foci in the same patient. This was possible when we analyzed a cohort of breast cancer patients where lumpectomies were processed using a whole mount technique. In these cases, the entire surgical specimen was sliced into 4 millimeters, and each slice was processed, allowing to pathologists to not only study the lesion, but also the tumor microenvironment. In this study that we did with the Ontario Institute of Cancer Research on Adaptive Oncology program, the TMA cores were taken from different regions of each focus of a total of 59 cases. The TMA was analyzed with multiplexing, and we found that when we compared the histo scores between different cores, as shown here, we observed a wide range of scores, especially for ER, in this case, L31. When all the cores were taken into calculation for a diversity score based on entropy, we found some lumpectomies show high levels for ER and or PR, suggesting higher levels of intratumoral heterogeneity in some cases compared to others. So now we can talk about immune phenotyping and spatial analysis that we looked at in this field. In recent years, there has been tremendous interest in single-cell phenotyping of the immune microenvironment in cancer, as the discovery of immunotherapy as a cancer treatment has gained significant impact. Even though we now learned a lot more about how this kind of therapy worked, there are still challenges in terms of the identification of the patient or the immune status of the tumor that may more likely respond to the therapy. As a result, there is still a lot of interest to identify particular immune cells that express PD-1 or PD-L1 checkpoint proteins, the activation or suppressive status of these cells, and where they are localized with respect to the cancer or with other immune cells. To answer these questions, we have been imaging protein multiplexing of these immune markers to study the immune status of different cancers and how they could change during treatment. With regards to the immune phenotyping, we took a very similar approach in binary cell counts by using cut points for each immune marker determined using control tissue. By doing that, we can determine the total number of CD3 lymphocytes, CD68 macrophages, and the cancer cells. Within each particular cell type, we can then study the fraction of cells expressing particular markers of interest. From the series of ovarian cancer sessions that we have looked at, we see huge differences in the cell counts. Some cancer cases can be considered as immune cold, while others are highly populated with lymphocytes. We also found some cases with high percentages of PD-1 positive CD8 cells, which has been suggested as good predictors to immunotherapy. We also use other methods to visualize data here, such as t-SNE, which is a high-dimensional embedding algorithm. We found that the composition of the immune cell in the cases can be quite different. When we annotate the expression of other functional proteins, in this case, Ki67, PD-1, and PD-L1 on these plots, we can identify the expression patterns on these immune cell types. Spatial analysis and cell arrangements are also the focus of our quantitative studies, as increasing evidence has suggested that the ways of the cells are localized in the lesion or around each other can have prognostic values. The proximal locations of these cells could also suggest that there could be functional interactions between particular cell types. Again, using neighborhood analysis approach, we studied the likelihood of different cell types or how the expression of protein signatures are in close proximity with each other. In this particular region of an ovarian cancer tissue, CD8 and CD4 cells appear to be in very similar cellular neighborhoods, as shown by the bar graphs here, while PD1 expressing CD4 and CD8 cells are quite different. CD8 PD1 cells appear to be more likely in close proximity to PDL1 expressing cancer cells. In this other region, when we compare the cellular neighborhoods of CD8 positive macrophages, it appeared that PDL1 expressing CD68 cells are more likely to associate with CD8 cells when compared to CD68 cells that are not expressing PDL1. We have also looked at the quantification of the actual physical distances, or the Euclidean distances between cells. Shown here is one of the examples where we studied the frequency of CD3, CD8 cells that are located at the indicated distance in pixels between each other, shown here on the upper left-hand panel. We also looked at the frequency that CD68 cells are located between each other at the indicated distance on the lower left-hand panel. From this, it looks like CD3, CD8 cells are more scattered in distribution, whereas CD68 cells are more uniformly located from each other. We also studied the distance between CD3, CD8, and CD68 cells as shown on the right. From here, it seems like CD3, CD8 cells are usually located to CD68 cells at close distances. In conclusion, we have used imaging protein multiplexing combined with quantitative analysis to phenotype individual cells in cancer and to also evaluate heterogeneity in both cancer and the surrounding tumor microenvironment. High levels of correlation of multiplex measured expression and IHC measured expression of biomarkers in breast cancers were detected, suggesting that the translatability of this kind of analysis to the clinical setting. Quantitative spatial analysis suggests that some cell types or cell clones are more likely to be in close proximity. We're actually expanding to use machine learning algorithms to identify these spatial patterns and how they could be used to predict outcomes in cancer. I would like to thank everyone in Dr. Martin Yaffe's group at Sunnybrook Research Institute, particularly the members in the Biomarker Imaging Research Lab, who has all contributed to the work that I presented today. Also, many thanks to our collaborators, and thank you to you for your attention.